Discovering new drugs, exploring the screening methods used by scientists. Drug screening methods are used to identify potential new drugs that can be used to treat diseases. These methods typically involve testing large numbers of compounds to identify those that are most effective at targeting a specific disease. There are several screening methods for drugs, including high-throughput screening, HTS. HTS is a method that involves testing thousands of compounds in a short amount of time to identify potential drug candidates. This is done using automated systems that can quickly screen large numbers of compounds against a specific target. Virtual screening. Virtual screening is a computer-based method that uses algorithms to identify potential drug candidates. This method involves screening large databases of compounds and identifying those that are most likely to be effective based on their molecular properties. In vitro screening. In vitro screening involves testing drugs on isolated cells or tissues in the laboratory. This method is useful for identifying the mechanism of action of a drug, as well as its potency and selectivity. In vivo screening. In vivo screening involves testing drugs in living organisms, typically animals. This method is useful for evaluating the safety and efficacy of a drug in a more complex biological system. Human clinical trials. Clinical trials involve testing drugs on human volunteers to evaluate their safety and efficacy. Clinical trials are the most important step in drug development and are necessary before a drug can be approved for use. Overall, a combination of these screening methods is typically used to identify potential drug candidates. This helps to ensure that only safe and effective drugs are brought to market.